Bien, a continuación vamos a abordar la temática Comportamiento de variedades tolerantes en zonas bananeras. El expositor, el doctor Eli Khayat, director científico en Rahan Meristem. Adelante, bienvenido.
Yes, in Israel, in the north side of the Western Galilee. And I'm speaking on behalf of Rahan Meristem, which is a plant biotech company in Israel. Okay, I'm going to speak to you about, I will try to validate the resistance that we are seeing in our GAL variety after manipulating it, but not by GMO or any kind of introduction of nucleic acid. So to start with, I would like just to list the different levels of validations that we have done until now, because I know that some people are doubting or have some questions about it. So the first level of validation we did when we used the mutagenesis type of diversification of a population of plants. After we prove, and I will show you evidence of each one of these points, after we proved the mutagenesis, we went to ex-vitro experiments where we used the expertise of the University of Wageningen at the laboratory of Professor Gert Kema and his colleagues. And we screened the population of mutants. And after screening the population of mutants, we identified plants that we called them asymptomatic at that point. There were approximately 415 plants from a population of 10,000 lines that we mutagenized. The next step, we went and performed a whole genome sequencing where we compared a resistant line to the wild type, and we looked at the differences, and especially in relation to relevant QTLs that we knew in advance that these QTLs reside in a position on the chromatin where there might be some resistant genes or genes that have to do either with the perception of the pathogen or the signal transduction or even the execution of resistance. After we've done that, we took the plants to a field trial. Actually, we used 3 different plantations in the Philippines that were heavily infected with TR4. And the last step, which is just these days, we moved the resistant lines after propagating them in a laboratory in the Philippines, then again in a laboratory in Israel. And now the plants are being prepared to be planted in the area of Colombia where it was found that there was an infection, the peninsula of La Guajira. So this field trial will start in Q1 of 2022. On the agro-technical parameters, we measured bunch weight, we measured the number of fingers, we measured cycling time. So these are the main components of yield. And we also looked at fruit quality in terms of proper ripening, the green life of the fruit, and Brix values. Okay, I would like to start my talk by showing something that most banana growers are very familiar with. And these are mutations that are caused by tissue culture. And these mutations are often in the literature, they are referred to as off types, or typically they're called mutations due to tissue culture, or there is another term of somaclonal variation. Okay, so the first one was a dwarf mutant. The second one is a very important mutation for our work, and it's called long and narrow leaves. I'm sure that many of the growers have seen them, especially in young plants. You see heavy pigmentation, and the leaf phenotype is different than the wild type. It's long and narrow. There is another common mutant, which is the expression of a pigment, a red pigment in the leaves. And there is variegation on the leaves. And it's again another mutant that it's not very common, but it's way above the expected rate of mutations if you look in a population randomly. Okay, the next mutation is a very interesting mutation. It's called Masada, that's how we call it, and some other, in other places they call it variegated mutants. And this mutation actually is interesting because the number of chromosomes, the N number is 34 instead of the 33, which is in the wild type. Very important. And also, if you look at the stomates underneath the leaf, you will see that the organization of the stomate, you can see on the left panel over here, you see that the stomates are not organized properly as they are in the wild type in straight rows. Okay, so this is the phenotype that you'll need some instrumentation to see it, but it's very easy to see the variegation on the leaf compared to a wild type. Okay, so the question is, why are these mutations appearing after tissue culture in such large numbers? What is the mechanism that creates these mutants, and why do these mutants repeat so many times? And at the end of the tissue culture, you may get a very large number of off types, but from very distinct groups, very distinct phenotypes that are not too many. So this is an interesting phenomenon in tissue cultured bananas as well as other plants. And again, I want to refer to the long and narrow leaf mutant that was the first mutant that we studied quite heavily. So we looked at when we started the population of plants in tissue culture, and after many, many cycles in the tissue culture, we performed AFLP analysis to look at polymorphism in the bending pattern of DNA. Now, all of these lines, all of these plants, they all originate from the same mother plant, but with many, many cycles of tissue culture, eventually you start seeing a bending pattern like you will see in lane number 5 that is different than the bending pattern of the siblings plants that were extracted exactly the same way and have the same history, if you wish. Okay, so the question was, when we started to work on this, what is this band, if we can sequence it and if we can position it in the chromatin and look what does it encode for. So we cloned this band, we took it out of the gel and we cloned it. And what we found is that it is really a very common sequence that appears in the genome many, many times. And the name of this sequence is Retrotransposable elements, or if you wish, just transposable elements. So if we look at the rate of mutations over tissue culture production, in bananas, we almost always notice that after probably 12 cycles or 11 or 12 cycles in tissue culture, you get a steep rise in the number of mutants, and around 14 cycles, it levels off, you don't get more mutations, and amazingly, you can get up to 60% of the plants that you produce as mutants. Now, what is the character of these mutations? We will see in a second. And the question is, what is the mechanism, the genetic mechanism for this mutagenesis? That was the question that we put. And looking at the gel that I showed on the left side, our conclusion is that it may be related to activation of retrotransposable elements. Now let's look at these elements for a second. Okay, so we know in nature, actually this is true for all eukaryotic organisms from yeast to humans, there are 3 types of transposable elements. Okay, our element is class 1, that's how it is referred to in the literature. There are class 2 elements and class 3 elements, which I will not go into details, but we discovered that it's not class 2 elements, the element that we are talking about, the element that we observed in the gel, that we sequenced. And it's also not type 3, but actually it is type 1 element. Now, how can we tell that it's type 1 element? First of all, by the sequence. And you see the sequence I will show a little bit later. There are very conserved genes and proteins that are produced from this element. It's actually, it resembles retroviruses that appear in different eukaryotic organisms. So these elements actually are transcribed into a polygene that can be divided into several proteins and a protease that will separate the different proteins, but it can go a different direction. It can, this sequence can undergo reverse transcription, and it will form a circular RNA that will eventually become, with the reverse transcriptase enzyme, will become a DNA molecule. And this molecule will migrate into, from the cytoplasm into the nucleus. And in the nucleus, it will reintegrate into the nucleus. Now, the interesting part is that the reintegration is actually, most of the time, it is in the old position where this element started from. Sometimes it goes to new positions, but that's very rare. In very, very high frequencies, it goes back to the same position where it started. Okay. So we wanted to validate that what we see really in the mutant, in the long and narrow leaf mutation, is really one of these elements. And one way to tell is by detecting a molecule of messenger RNA. And here you see a PCR gel. See, this is the DNA. So in all the plants that we looked before, we detected the DNA because it's part of the genome. But in some of the plants, by using RT enzyme or by omitting the RT enzyme, you can prove that these plants, these mutants, actually activated a retrotransposable element because we see the RNA of this element. And if you sequence this RNA transcript or the cDNA, you will find out that it is part of a retro element. So I think that doing this, we more or less proved that what we are doing, the mechanism that causes this mutation, is basically a mechanism of activation of retrotransposable elements. And by doing so, we get mutants. Now, if we perform what we call a DNA blot or southern blot analysis, and we can see in lane 5 and 6, these are the wild type lanes, and we use a small segment from the retrotransposable element. Actually, it's a part of the RT enzyme as a probe, and we hybridize the probe to the DNA blot. But we see that in some of the bands that pre-existed in the original wild type, we see a very strong amplification of the same band. And we can see some bands that are undetectable here. Maybe they do exist or not, we are not so sure. But we definitely see that some of the bands that existed here are very strongly amplified here. This proves that the retro element mainly returned to the same position where it was when it started, before it underwent transcription. So again, that's a proof that the mechanism that we are talking about is really activation of retrotransposable element. And we used the rice reverse transcriptase homolog or ortholog as a probe. Okay, next step. Of course, very quickly we went and got a patent on this technology as a technology for creating diversity in bananas where there are no seeds, and so the diversity is very limited. But we know that this way we can create a diversity, and I will show how we can activate it even stronger, or we can use tools, genetic tools, to manipulate the plant and activate a large number of retrotransposable elements. So basically, our technology entails producing a library of mutants by activating these retrotransposable elements. Now, what's so nice about these mutations is that these mutations are very mild. So basically, what we get is a very small change in phenotype. Why? Because we're not hitting any structural genes or any kind of genes. We are in every instance that we looked at, we're hitting spaces between active genes, and the changes that occur in most cases, and this is our hypothesis, is really what we do is we change the conformation of the chromatin. By changing the conformation of the chromatin, actually now we have tools to see it actively. So we are looking at that. But by changing the conformation of the chromatin, we exposed different parts of the chromatin that were entangled before, and they were not exposed to all types of factors that are responsible for transcription. We may also hide some portions of the chromatin. So if before these portions were exposed, now they're hidden, and by that, we are causing these mutations. Now, of course, these mutations are very stable because the retrotransposable elements are real elements that integrate into the genome, and they don't come out of the genome. And we have some examples where we've done this on different genes in banana plants, and we got very stable mutations over even 15 years. Okay, now let's go to the TR4 resistance, and I'll let you guys judge if it is resistance or tolerance, okay, to the disease. Okay, so this is how we designed the experiment. We started with a very good clone, our best variety. It's called Gal. It was proven in many parts of the world as an excellent variety, high yield, excellent return, some stress tolerance, and so on and so forth. And we performed the in vitro mutagenesis on these Gal mutations. And we created 3 populations, but the populations that were created here, they were created with the last cycle of the tissue culture. In the last cycle of the tissue culture, we got a clump that in the previous cycle was a single plant. So basically, these are siblings from a single cycle of a single plant, each one. And so each one of these populations, we have 3 populations, is they are very similar to each other. And these populations represent each a single clone that was in the previous cycle of the tissue culture. It was a single clump. Okay, once we did this, okay, we took one subset of the population and we sent it to the University of Wageningen for screening. Now, each one of these, it's approximately 10,000 plants, but each one of the plants, to remind you, has 2 siblings, which are supposed to be very similar to this plant unless the mutation occurred at the very last cycle of the tissue culture, which is very unlikely because we used a very large number of cyclings. Okay, and so we did the selection at the University of Wageningen under greenhouse conditions with strong inoculations with the pathogen, different races of the pathogen. So we used really the expert domain of the University of Wageningen, of Professor Gert Kema's laboratory. Once we got the selected plants that showed no symptoms, we call them asymptomatic lines, we sent these, we first propagated each one of these lines, and we sent them to the Philippines to be tested under severe TR4 conditions or fields that have a very, very high inoculum of TR4. Okay, so just to summarize until here, okay, we had the germplasm, that we selected and we mutagenized. We did a selection and validation. And the last part, we are using a field evaluation in order to validate that everything that we found is really will withstand the pressure of the disease in the field. Okay, so here I'm showing the DNA banding pattern, and what we have discovered is that we can activate these retrotransposable elements with very high frequencies if we demethylate the DNA, because in most cases, actually in almost all cases, except for very rare mutations, these retrotransposable elements in the genome, they are inhibited from transcription by methylation of their promoters. So by demethylation, we activate these retrotransposable elements. Of course, we can deactivate some other components of the genome, but basically these are become very active. And you can see this is pre-activation and this is post-activation. And you can see the new bands, just like we got in the previous gel that I mentioned on the previous Southern blot. So this is a Southern blot again, using the same probe. And you can see the amplification of some of the bands and the changes in the chromatin, basically beautiful mutagenesis just by activation or demethylation, which activates these retrotransposable elements. Okay, so the next step of our work was bringing the plants. This is Dr. Fernando Garcia, who was at the time a PhD student at Dr. Kema's lab. And we came and we gave them the mutated lines. They performed beautiful hardening and potting of the plants. And approximately at about, I would say about 35 to 40 centimeters tall, the plants were inoculated with the disease. And we had two batches that we started with. One batch was 4,500 plants. We discovered that 153 plants were asymptomatic. On the second batch, we had 5,200 plants. Fernando was really very diligent at this, and he found 262 plants, which is approximately 5% of the population was asymptomatic. So in total, we got 415 selected lines that were at this point asymptomatic. So these lines were, of course, repropagated in tissue culture. But before we did this, we wanted to make sure, we wanted to validate the screening system. So the way we validated the screening system, actually, this was done by Fernando, and it's part of his PhD project. So we sent Fernando a line of Cavendish that is called ADI, another line of Cavendish, which is the Granane, and Gros Michel we used, that was propagated also by us. And we inoculated these first with Fusarium oxysporum race 1. And you can see, as expected, the Gros Michel showed strong symptoms, while the two Cavendish varieties did not show any symptoms. If we just used water, of course, we did not get any symptoms on any of the varieties. And when we inoculated the plants with TR4, of course, as expected, all of the varieties showed very strong symptoms. So this, to me, was a good screening system. It works just as expected. And so we trusted the screening system and we continued from then on. Okay, here's some photos of the clean plants that showed no symptoms. Actually, this is very interesting. This pathologists will call these type 2 resistance, where the pathogen actually tried to penetrate, but was somehow stopped by the plant and did not progress inside the plant. But here you can see that the plants were completely white. They did not show any symptoms whatsoever. Okay, so this is the first batch of from 3,947 plants that survived this test. 153 plants showed either resistance or tolerance, or at the least asymptomatic phenotype. Okay, so we took the plants and we propagated them, and all of the lines, all 415 lines, and we produced many exemplars from each line, and we introduced all these plants into the field, and we looked in the field. After the first cycle, 99.5% of the first 3,000 plants from the first batch were asymptomatic in the field. And on the second plot, we had 4,500 plants, and here we got 96.9% of the plants did not show any symptoms. So this was a very exciting moment, because it shows that really the selection that was performed at the University of Wageningen actually worked well. Oh, sorry. Okay, so we continued with this experiment, another 3 cycles of fruiting. Actually, the plants are now in the field in their 4th year, okay? And over the 4th year, over 4 years, we selected approximately more or less about 20 lines or 22 lines that were completely asymptomatic, and we call asymptomatic or resistant line only a line that if we used 120 plants from that line, not even one plant was infected in the field. Not a single plant to be referred to as a resistant line, not a single plant from the entire line of 120 plants would be infected in the field. Now, this is the situation that we saw in the field. So this is one of our best lines. It's called 8794. And you can see that all the plants around it, okay, the red dots represent plants that were infected and either died or showed very heavy symptoms. And the 8794 did not show any symptoms. And the bunch was absolutely marketable. There was no difference between this bunch and the Gal variety, which I will show soon. Okay, and we had, like I said, 22 of these lines, but we, from the 22 lines, we are propagating all the 22 lines, but we selected 6 lines that on the agronomic level, they performed much better than the other lines. So some of the lines had maybe a little bit less bunch weight, or the bunch weight was a little bit lower, or they had less fingers. But you can see, for example, this line looks like the bunch is beautiful, and there is really nothing wrong with this plant. And this is after 4 years in the infected field, where you can see that the most majority of the plants around it, okay, all died, and this one really survived. Okay, so it's very important to us as a commercial company not to just rely on resistance, but to measure all other parameters, like I mentioned before, that are important to the growers. So, for example, and we compared it to a normal gall, okay, a gall that was grown in areas where it was not infected with the disease. So, for example, if we take bunch weight, you can see that 8171, the bunch was pretty much the same as, and especially 8794, pretty much the same as a normal gall. There is really no significant difference statistically. They are similar. So we are very happy that the plants really don't show any strange phenotypes. And like I mentioned before, the mutations that we create by exposing certain chromatin areas are very, very mild mutations, and we would not expect major phenotypes that will be deleterious or would be disadvantage to the grower. So here we show the cycling time, which is again a very important component of yield. And we see again that really basically we don't have any significant difference between the gall and the other 2 lines. And we can go on and show that 6 lines, actually some of them in some of the parameters overperform the normal gall. So after this, we went and performed a whole genome sequencing as the next step, or the next level of confirmation. And this is the sequencing statistics. I'm not sure if we should bother with it. It's just for the bioinformatics. But up here is a very important piece of data. What we've seen here is that there is one landing pad or one area where these retrotransposable elements reintegrated into the genome, which is in very close proximity to the RGA genes, which has been proven to conform resistance to the disease. Now, we did not change anything on the RGA gene. We did not modulate it. We just have these retrotransposable elements land in a close proximity to this gene. It's approximately 500 kilobase pairs away between the last retro element at the 5' side of the sequence and the RGA gene. Okay, I will move on. So this was, again, this is for us a good way to confirm that what we have here is really a mechanism that was created by these retrotransposable elements where they reintegrated into the genome in a specific locus where they are going to have some kind of an influence on the transcription of this gene without modulating the gene itself. And again, we're not using any GMO or which I have nothing against it, but we just, as a commercial company, we were very careful to use techniques that will be acceptable to all, and we can market our products. So finally, this is some data that shows that simply the polymorphic loci between the demethylated plant, one of the lines, and the wild type is approximately 12,270. It's not unusual. If we take two plants that were produced from the same mother plant in tissue culture, you would probably get a similar figure. It does not represent a very high rate of mutated nucleotides. The other thing that we saw is that some of these retrotransposable elements really are concentrated in a region in the genome where there is a cluster of R genes, and we discovered that. And here there could be, because there are several alleles to each of these genes, but altogether we found by our bioinformaticists that are 464 I would say mutations near R genes, not inside R genes, but near R genes. So that's quite heavy, and we don't know if this is a preference to the retrotransposable elements. This was created somehow by evolution or something like that, but it seems to us that it is really a high frequency of concentration of retrotransposable elements in the vicinity of several of a cluster of R genes. Okay, now we are in a position where the plants were repropagated and were sent to Colombia. We got the permit from ICA to import these plants to Colombia. They will be planted in La Guajira, as I said before, in the first quarter of 2022. And what we will want to see is to confirm again that these plants are really, they merit the notation of resistant plants. So we are doing, and by the way, we have another experiment in the Philippines where there are some of these 6 lines, there are 60,000 plants, and it's already been more than a year that they are planted in the field, and the rate of symptoms or symptomatic plants is less than 1%, over 60,000 plants. So to me, I'm confident as much as I can be that these lines represent a population of mutants that are resistant to the disease. I would say resistant because of 4 cycles in the field. Seems to me like it's quite stable. And so from here on, we will just wait for the experiment in La Guajira, and hopefully they will show the same pattern that we have seen in the Philippines. So at this point, I would like to thank everybody, and especially to thank the people that supported us and gave us, assisted us in this research. And I would light up and just would like to thank very much Mr. Luis Alberto Restrepo from Banarica in Colombia, who financed this work together with Rahan, and to our NRGene, who did the sequencing and the analysis of the sequence data, and of course Professor Kema and Fernando, and one more person is Lalin, who is the grower in the field where we performed 2 field trials in Dabong. It's in Valencia City, Bukidnon in the Philippines. Thank you very much. If there are any questions, I'll be very happy to answer.
Bien, si acaso tienen preguntas, pueden levantar la mano para poderles llevar el micrófono y que puedan formularla.
Por favor, en inglés.
Okay, no question.
No question.
Yeah. Okay, sí.
Everything
yes.
is clear.
One question.
Okay.
Good afternoon, doctor. The bananas that are going to be planted in Colombia in 2022 are going to be just one species, or you're going to send the different ones?
No, I'm going to send, we already sent all 32 clones that shows no symptoms at all, actually a little bit more than what I mentioned before, but we have 6 preferred clones that we will focus on them, but we will send all of the clones. By the way, we planned a similar experiment in Ecuador. We already have a permit to import the plants to Ecuador, but in Ecuador there is no TR4 as far as I know, and so we cannot look at the resistance, but you will be able to look at the performance of the plant agronomically.
Okay, thank you, doctor.
You're welcome. Excellent question. Okay, so the plants, yes. So the plants traveled, from the Philippines to Israel. And in Israel, we sampled the plants. There is no bunchy top, and there are no other diseases, no banana diseases, especially, or nothing that we can measure, okay? But ICA, and of course, the same thing will be performed in Ecuador by Agrocalidad, will take samples, and they will look at all of the other diseases, because we don't want to import, and so the plants will be in quarantine until we are clear that by ICA and Agrocalidad that the plants are absolutely free of all plant viruses and diseases. And we already did these tests in Israel, so we know that at least the clones that we examined, we know that they are free from diseases and any kind of virus, bunchy top, and even CMV and other viruses, other banana viruses. No viruses. okay, and no diseases. Diseases, it's of course, the plants will not have any banana diseases, not moco and not fungi, because if they did have these, it will be expressed on the medium. They came in vitro to the countries in Latin America. So, but there are no diseases, but nevertheless, they will be tested in each one of the countries that we will export the plants. And I'm certain that they will not find any diseases, but we are happy to give a chance to the authorities to make these measurements.
Dr. Eli, well, you already answered my question, but I would like you to elaborate a little bit more about the testing of bunchy top banana virus. How much time do you wait until you test for the virus on Israel after bringing the banana from Philippines? You know, bunchy top can express after even 1 year. And when there's planglets, you cannot detect those. So I would like to understand how long do you wait until the testing?
Okay, so also in Israel, unfortunately, by the way, bunchy top you can detect in the greenhouse. I've seen those many of these in India and in Africa. You know, it is possible to detect them even visually in the greenhouse. Of course, if you do a real-time PCR analysis, you will detect them. But anyways, in Israel, we could not take them out of the greenhouse because of quarantine. So they were only detected in a greenhouse. So it's not years of growth. It's just a couple of months. Okay, but I would like the authorities in Agrosavia and ICA to have the opportunity to test them with your own tools and your own expertise and verify that they are really clean. To me, they are absolutely clean, but I would like the authorities to confirm.
Bien. Muchísimas gracias a nuestro expositor, el doctor Khayat.